This video features one of Anogen's best-selling products, the Multiplex Human Cytokine ELISA kit. Non-functional pituitary adenomas, or NFPAs, are among the most common primary brain tumors. Although benign, they are space-occupying masses which compress parts of the brain, causing hyperpituitarism, vision loss, and debilitating headaches. Previous studies have highlighted the role of tumor-associated macrophages, or TAMs, in the initiation and progression of tumors. This is because tumors are capable of hijacking macrophages and converting them from default type M1 anti-tumoral phenotypes into type M2 pro-tumoral phenotypes, which are capable of promoting tumor growth and invasion. A study published in Oncotarget stratified NFPAs into two groups, distinguished by these two macrophage subtypes. The study found that NFPAs with more TAMs were the most expansile. NFPAs without cavernous sinus invasion had more M1 anti-tumoral TAMs, whereas NFPAs with cavernous sinus invasion had more M2 pro-tumoral TAMs. In cultures, M2 macrophages promoted greater invasion and proliferation of primary NFPAs than M1 macrophages. Findings from this study will help generate immunomodulatory therapies for NFPAs. Anogen's Multiplex Human Cytokine ELISA kit was used in this study to assess M1 and M2 macrophage polarization. Anogen supports researchers in cancer immunotherapy and tumor microenvironment by providing hundreds of high-quality antibodies and ELISA kits. For more information, please visit Anogen's website at www.anogen.ca.